Humans have long been the dominant species on this planet. Our ability to develop technology grows more and more extensive every day. We have engineered things that were long considered impossible, like self-driving cars and robots. But recently, scientists have discovered how to genetically modify organisms. They found a way to go into our DNA and change its structure to target traits that we deem bad, and that technology is known as CRISPR, or Clustered Regularly Interspaced Short Palindromic Repeats. It allows humans to edit genes by targeting specific DNA sequences and using the Cas9 protein to cut the DNA. This allows a new DNA sequence to be added to a specific gene. What does that mean? It means taking out the gene that causes many genetic disorders, like albinism and Down syndrome. Yet, it is not limited to just disorders. It could allow for excessive strength or blonde hair. The possibilities are endless. In the early stages of CRISPR, scientists were excited to hear about a new technology that might be capable of sustaining biodiversity. Researchers wondered whether or not CRISPR could be used to preserve coral reefs and other ecosystems facing biodiversity loss. While the technology can potentially intervene in active extinction and prevent the loss of an entire species, many scientists have reservations, noting that there's no way to ensure the results of human intervention in the natural flow of evolution. There is no doubt that the presence of human life on this planet has greatly impacted millions of species across the globe. However, the effects of further involvement are only sometimes predictable. There is always the possibility that we make things worse. According to Tony Piaggio, a research scientist for the USDA, CRISPR can be catastrophic, and scientists should only partially indulge in its capabilities.
Well, I think we should use or consider using all the tools we have at our disposal to help species not go extinct. There are a lot of things that we have traditionally done, like habitat protection and intervening in the killing of animals or plants. But CRISPR offers a brand new way to address some of the underlying genetic problems that some species have. So a lot of species, when they get to small population sizes, they get things like inbreeding depression and other problems associated with not having very many individuals in the population. And so bad genes come to the forefront. And if we could use CRISPR to address some of those bad genes, then that might help them stick around a little bit longer. There's always concern about changing species' genetic makeup to help, in this case, conserve them. Is a species still the same species if, for example, you insert some new genes into their genome that come from another species, perhaps? Would you still consider that the same species, or is it something different? And so now, instead of protecting the thing that you were trying to protect, you've actually created something new and different.
So you feel since the technology is available, why not use it?
It should be, in my opinion, one of the tools we have to help try to manage and conserve species, yes.
Coral reef ecosystems are one of many that are facing biodiversity loss. Tony Piaggio goes on to say that scientists intend to use CRISPR to understand, in a laboratory setting, which coral reefs are important for handling stress in the environment, and use that information to help other coral conservation efforts. They aren't just messing around with their technology to make heat-resistant coral. The proof of concept study was successful, confirming that the technology can be applied to the ecological conservation field. An article published in the CRISPR journal conveys the idea that with the increasing number of wildlife populations seriously impacted by exotic diseases, invasive species, climate change, and missing ecological processes, there is a need to expand the conservationist's toolbox. CRISPR-Cas9 genome editing offers a precise and potentially transformative tool to confront these challenges. In the past 23 years, the number of species facing extinction has increased dramatically. Without CRISPR, the number may continue to increase, eventually leading to a mass extinction.
So I have mixed feelings about this because I think that if you're going to do that kind of thing for anything, that's a good cause. And I would be supportive of something that could help with endangered species in that kind of way. So that's one side of it. The other side of it, though, is concerns about not knowing what all of the impacts of it might be. There might be parts of the ecosystem, parts of the scenario that you're introducing it to that are affected in ways that we can't predict. So I would just have concerns about how well we could know or predict that there wouldn't be significant negative impacts in ways that are hard to fix. And then the other part is, I think, the possibility of it being used in ways that aren't good, or I guess you could say misused. And if it's used for endangered species, that's one thing, but if that opens things up for it to be used in other ways, or for people to find ways to manipulate things, whether it's regulatory aspects of it, to get around any laws or things that would keep you from misusing it, they might find ways to get around it and do things that are dangerous or harmful or generally not good or ethical.
What risks do you think will come from intervention in the future?
So the first thing that comes to mind is the difficulty with creating a situation that you can test things out and try to figure out how things will interact once you introduce it into nature. I think that's there's a lot of limitations with what you can do in a controlled setting. I don't know how exactly they would be doing that, but the other piece is, seeing what will happen and how things will interact over time and respond over time is probably important. Kind of just the complexity of nature that wouldn't be necessarily present in the controlled setting that you're studying beforehand, and the limitation on time to see how things will respond. Both of those make it difficult to know exactly how it will impact things in nature once you actually do it. And so I think there's a risk there that there's a lot of unknowns.
A factor we need to consider when using CRISPR is that we are creating genetically modified organisms, which have many associated issues. We are interfering with the traits present in nature, and we need to acknowledge that these actions may have implications in the future. Genetically modifying an organism using CRISPR may decrease the genetic variation of that certain species or population. While in the short term, these organisms may survive with certain challenges that CRISPR eliminates, this might lead to the species not being able to adapt to new challenges in the long run. Not only can CRISPR cause negative effects to a certain species, it can affect the entire surrounding environment and ecosystem. Genetically modified organisms can be associated with the use of chemicals and certain substances leached into the surrounding environment. This can lead to contamination of soil and water, creating chemical toxicity. The soil becomes less fertile, and contaminated water causes a decrease in marine productivity, ultimately decreasing the biodiversity in the ecosystem. The loss of a keystone species can be devastating for an entire ecosystem. Coral reefs are a great example of a species fighting a losing battle against man-made biological catastrophes. We have gained a new scientific power unlike anything we've ever seen before, with the ability to save species from extinction altogether. We've dedicated decades to find ways to more effectively conserve endangered species. CRISPR has challenged our former conservation efforts. Rather than change an entire environment, we can edit a specific species so they're more resistant. While we have identified the possible consequences of using CRISPR in active extinction, they don't outweigh the benefits. Without the use of CRISPR, many species could go extinct in the next decade. If we use this technology to save species responsibly, and only through regulated groups, we can use this novel technology to change the future of our planet for the better. So, what's stopping us from intervening and genetically modifying organisms to prevent their extinction?